All right, so with acids and bases, recall that when you have two water molecules interacting with each other through hydrogen bonds, at any point in time, the hydrogen involved in that hydrogen bond can dissociate from one of the two water molecules, resulting in the formation of what's commonly called a hydroxide ion and this guy, which is commonly called a hydronium ion, which is usually represented as a proton. Okay. Now, as long as we have an equal number of hydroxide ions to hydronium ions, or protons, we can always go back to having pure water. Hopefully everybody kind of sees that. And as long as those two concentrations are equal, we can always go back to having pure water. But there are certain solutions that can mess with this concentration. And because these two are related, based off of the dissociation of a water molecule, when we increase one, we decrease the other. So if we increase the proton concentration, in response to that, we're going to decrease the hydroxide ion concentration. And there are names for the solutions that manipulate these relative concentrations. If you are messing with the hydroxide ion, the hydronium ion or proton concentration, you're going to be looking at an acid. So an acid increases the proton concentration and it decreases the hydroxide ion concentration. Whereas if you're looking at a base is a substance that decreases the proton concentration but increases the hydroxide ion concentration. Okay? So this is the difference between acids and bases. Acids increase the proton concentration and decrease the hydroxide ion concentration, and bases decrease the proton concentration and increase the hydroxide ion concentration. Okay? I guess I could probably do the number up here. There we go. Yes, increase the hydroxide ion concentration. It doesn't decrease both of those. Just understand, if one is decreasing, the other has to be increasing. Yes, sir?
Say it one more time.
Absolutely. An acid is a solution that increases the proton concentration but decreases the hydroxide ion concentration. A base is a substance that decreases the proton concentration but increases the hydroxide ion concentration. Okay? Now, in the lab, you focus on one of these two ions. You focus on the relative level of protons inside of the solution. And to measure that, the scale that you used is commonly called the pH scale. So for the pH scale, what we're really looking at is how acidic or how basic a solution is. And we'll also think about what that means in the context of protons and hydroxide ions. So pH measures the proton concentration. If you have a pH that is less than 7, you have a solution that is high in proton concentration, low in hydroxide ion concentration, and we classify this solution as an acid. If you have a pH that is greater than 7, you have a low proton concentration, a high hydroxide ion concentration, and you have a solution that is classified as a base. And then finally, if you have a pH equal to 7, then you have a proton concentration that equals your hydroxide ion concentration, and you classify the solution as neutral. Now there were 3 different techniques that we used in the lab to measure pH. So when we're talking about pH measurement, we're talking about 3 different techniques. What was one of the 3 techniques that we used to determine pH? We used the pH meter. Okay. Yes, we used the pH paper.
And start with the A, I think.
The colorimetric, the cabbage juice, which is commonly called anthocyanins. I have a lot of students in my lab ask me, do I count off for spelling? Now, for my lab, I will say this, for my students, as long as it's maybe 1 or 2 letters difference, I'm probably going to give you full credit. I can't speak for other instructors. Other instructors might be more or less strict in regards to their grades on spelling. I will tell you what will not earn you any points, though. My students already know this. Is if, let's say, that you can't remember that it's anthocyanins, but you remember the first part, a lot of students like to do this. Right? We understand that if you just write the first part, you can't remember the second part. We don't think that somehow this magically translates to science. Okay? So please make sure that you're comfortable with spelling what you need to spell.
So
Yes?
is it the same thing as saying a set of standards?
What's that?
Is that different than just saying a set of standards?
Yes, that is different. Anthocyanins are the specific molecule. There are a variety of different pH indicators. You have to say anthocyanins if they ask you about that. And cabbage juice might not even be necessarily acceptable. So definitely I would know
So you put it in
anthocyanins.
Spanish?
What? I cannot.
As I usually do, that I can write.
Alright. Now, of these, which was the most precise? Which of them gave us the most precise measurement of the pH?
The meter.
The meter, right? Because it actually gave us a pH that had tenths and hundredths places in comparison to the anthocyanins and pH paper, where we had to use scales and we only got those values in whole numbers. Okay? So that's really what you did, was you just measured unknowns. I would say also knowing the color spectrum for the anthocyanins is a good thing because you build this whole scale. Anybody remember what colors were typically acidic?
Is it green?
The pink and the red, okay, whatever. If you write hot pink or bubblegum pink, they're not going to count off for that because you still understand that pink means acidic. What colors did you typically see for your bases? Blue and I believe a green that might have eventually faded to like a grassy green or a yellow, right? Okay. So, I have videos and images again. Go study them if you need to, just so that you're aware. Alright, so, outside of just measuring the unknowns, you also measured household solutions. They're probably not going to ask you to memorize the pH of each household solution because guess what? Each group probably got a different pH. Okay, so I would say knowing how to use these different techniques is probably beneficial. And then knowing the color ranges, probably good for you as well. Okay. Yes, ma'am?
You said it's good for us, or do we have to, is it like mandatory to know it?
That I don't know. It's going to depend on what they choose to ask.
Oh, okay.
But it can't hurt you to be more knowledgeable than less knowledgeable. That's kind of my whole mantra. Yes, ma'am? What color is neutral with these? I believe in the anthocyanins case, it was more of kind of like a gray, possibly blue steel color. Okay, that kind of had this off, almost like a slight purple, but not really. Okay. Yes, ma'am?
I thought all the practical tests were going to be the same.
They might not be. There is no guarantee that they're going to be the same. The department chairs write the exams, and I know that there are multitudes of questions that can be swapped out at any particular point in time. So do not assume that just because you went and took the test or your friend went and took the test, that you're going to get the exact same questions as them. Yes, ma'am? They are timed. You get 90 seconds per station. Okay, there are 50 different questions over 24 stations. I'll tell you right now, most of the questions can be answered in 5 words or less. So if you're writing the great American novel, you're doing wrong. Okay? Now, what I do at the end of mine, after all the students have moved through all 24 stations, I do let them go back through and answer things they may have missed. Again, instructor by instructor basis. Okay? If you know your stuff, You're going to answer the question quickly. It's going to be the longest 90 seconds of your life. If you don't know the answer, you're going to sit there and wallow in misery, and it's going to be the longest 90 seconds of your lives. So, real quick, let's close up acids and bases with the final experiment you guys did, and then we're going to jump into macromolecules, and I think we're going to finish maybe either on time or just a little bit late, but that's okay. The final exercise you did was the buffering exercise. Remember that buffers are solutions that maintain their initial pH in the presence of an acid or a base. What this means is that whatever the buffer's starting pH is, if it's a good buffer, it's going to retain that starting pH even when you add an acid or a base. Doesn't have to be a pH of 7. If the buffer starts with a pH of 4, and I add an acid or a base, and it's a good buffer, guess what its pH is going to be? 4. Okay? If it's a good buffer, and its pH is 11, and I add an acid or a base, guess what? It's probably still going to be 11. Okay? So, when you guys did this, you had two solutions, A and B, and the question you were wanting to answer was, which of these two solutions has the greatest buffering capacity? Now, what those words mean, buffering capacity, it's asking the capacity of the solution to act as a buffer. In other words, how good of a buffer is it? And it's a comparative term. So you don't just say, this solution has a great buffering capacity. You say, it has a greater buffering capacity than this other solution. Okay? For my students in lecture, this was the example I gave where you could say about me, Professor Garcia is shorter. Okay? Well, shorter than what? You have to compare me to something. I know it can pretty much be anything at this point, but you have to compare my height to something. So you have to say, Professor Garcia is shorter than everybody. And then that is a factual statement. Okay? But you don't just say Professor Garcia is shorter. You compare me to something, and the same is true with buffering capacity. Alright, so we started with 0 milliliters of a substance, and in one direction we added milliliters of hydrochloric acid, and in the other direction we added milliliters of sodium hydroxide. As the names imply, hydrochloric acid, that's an acid, so we're adding more acid. Sodium hydroxide, NaOH, that's a base, so we're adding more base. And then what we did was we had our two solutions, and we were looking at their relative pHs. Now in our particular instance, I believe both of your solutions started with an initial pH of 7. So let's say that solution A is in black, and then solution B is going to be in purple. Now let's assume that solution A does this. Okay? And then let's assume that solution B does this. Which is the better buffer, A or B? A. Why is it not B? Because remember, the definition is it maintains its initial pH for a longer period of time. Notice that A stays almost entirely flat for a really good amount of time with the addition of either acid or base, while B skyrockets or plummets almost immediately. So in this particular instance, I would say A has the greatest buffering capacity compared to B. Or B has the worst buffering capacity compared to A. But if all of a sudden I had another solution, solution C, which ironically enough might be hard to see, and it did this, where it just stays straight the entire time, now which solution has the highest buffering capacity? C. Whereas A is greater than B, but worse than C, and B is worse than both A and C. So does everybody understand how you use buffering capacity?
Yes.
It's comparative. Okay, awesome. So that winds up acids and bases.